Hello and welcome to Invertebrates Part 2. This will be a general survey of a lot of invertebrate phyla. And we've already learned about Cnidaria and Platyhelminthes and Rotifera or Syndromata, as well as Brachiopoda and Cyclophora. So, now we're on to Mollusca, Annelida, Nematoda, and Arthropoda. And we're going to start off, amazingly enough, with mollusca. So mollusks are extremely diverse, right? We're talking snails, clams, mussels, octopus, squid, right? Cuttlefish. Also a weird little group called chitons. Right, there's a lot of animals in here. And they are extremely diverse in morphology as well as habitat, right? So octopus, squid, and cuttlefish, they're cephalopods, and they can be found in the deepest abyssal zones of the ocean. Colossal squids live 7,000 meters deep. And then you can find snails up into the alpine environments above tree line. So we're talking nearly every environment, talking nearly every elevation, polar seas, open ocean, tropical through polar climates. It's crazy. So with all this diversity, that means they're second only to arthropods for the number of species. Second only to arthropods. I keep leaving this plus 664 in because a ferret who passed years and years ago once ran across my keyboard while I was editing this lecture and added plus 664. And what can I say? I've got some, empathy and sympathy and whatnot. So, second only to arthropods in diversity. And then they have significant economic impact, whether that's positive or negative, right? In the world of human use, jewelry, right? Pearls come from clams. In the food industry, clams, mussels, squids, those are huge food animals. In the environment, some of their negative economic impacts, the zebra mussel. The zebra mussel is an insane invasive species. So it is not native to the U.S. and it is highly invasive in the U.S. Basically, wherever zebra mussels manage to colonize, they essentially, let's mute that. Wherever they manage to colonize, they essentially turn every exposed surface into a pure clump of massive amounts of zebra mussels. They don't have any predators in North America. Nothing has even adapted to eating them. So they just spin out of control and destroy the environment. They take all the nutrients out of the water and they turn it into like a dead zone filled with zebra mussels. It's gross. There are things like electrified portions of water near harbors to try and keep zebra mussels from getting into bodies of water like rivers systems. So there's lots of stuff going on. In the state of Colorado, if they find you with zebra mussels, you'll be paying a hefty fine, even if they're just encrusted on your boat because you vacation somewhere. So, and then of course, another big economic impact that's sort of on the negative end, Snails are often a intermediate host for platyhelminthes parasites. So intermediate host for platyhelminth parasites. Mollusks, big deal. Okay, continuing forward. Let's talk about the generalized body plan of the mollusk. So all mollusks have some degree of what we call a muscular foot. So the muscular foot can do a number of things, right? It can be involved in locomotion. Snails use their muscular foot to move. So snails will secrete a slime on the bottom of their muscular foot that reduces friction and allows them to scoot along their slime. So that's locomotory. Sometimes they're used for attachment. So if we just erase this right here, this clam has a muscular foot here that is used for attachment, digs into the substrate and holds it on there, and actually can be used for emergency movement as well. So that's kind of cool. Lots of cool stuff going on with the muscular foot. The chitons even use their muscular foot like a suction cup. So lots of functions here. Locomotion. Whoa, that is, let me try and write that a little nicer for the next ones. Locomotion, attachment, even suction for the chitons. That is supposed to be locomotion. All right, then we have the visceral mass. So the visceral mass is a mass of tissues. Essentially, a mass of tissues essentially, this is area where in most other organisms, we'd find a coelom. There is a coelom in Mollusca. Mollusca are coelomates, right? They have a coelom, but not all of their internal organs are present in the coelom. Our internal organs are, all our abdominal and chest organs, they are all present in the coelom. Whereas in Mollusca, there is a visceral mass where a number of organs that would otherwise be in a coelom in other organisms, other phyla, are not present. They're stuck in a mass. So this mass of tissues, it has the digestive system, most of the digestive system. It has, the location of the stomach and the anus. Right? It has a opening to the circulatory system. So let's erase this here. So the circulatory system does open up into the visceral mass. It's not completely solid. The tissues are porous in here. It's just not a hollow body cavity. Massive tissues, porous, but no body cavity. No body cavity in the mass. So no body cavity in the visceral mass. Still a body cavity, just not present in the visceral mass. Okay, circulatory system, right? We have an open or incomplete circulatory system. Boop, 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 screen. White screen. Okay, so here's the heart. Here's a respiratory structure. Could be gills or lungs. Right? And then the heart is going to have just some basic open vessels. Right? So these open into the body cavity. So they open into the visceral mass in the case of mollusks specifically. So which is that porous tissue. And basically, the visceral mass consists of a large, it's porous, so it also functions as a blood sinus. So functions as a blood sinus because it's so porous. So basically, rather than so this is an open or an incomplete circulatory system, whereas a closed circulatory system is one like mammals have, right? So you're going to have vessels, arteries, capillaries, veins, the heart. Everything is fully enclosed. It's in a closed circuit. Blood is not just being sloshed around and sucked back into a heart. It all travels in these one-way systems. So here's respiration, here's body, here's the heart. Right, so this would be a mammal closed circulatory system, or complete. So as we can see back in our picture, simplified, but we have a vessel that just pumps blood loose, sloshing around in this porous visceral mass. And it just sort of returns it's just sort of a pump in and out type thing. It's very simple. It does not have a complete return system. It's very size limiting. Most mollusks that have an incomplete circulatory system, an open circulatory system, are highly size limited. So it's not as bad as, waste and gases and nutrients being distributed through the body purely through diffusion, but it's not efficient enough to get truly large megafauna. You can get surprisingly big when you're in the ocean, though, but that's an aside. Okay, continuing on. The mantle. So the mantle is a sheath around the visceral mass. So if we go and make a new picture, white screen. So here's my visceral mass. And then I will have a mantle. So this is the mantle, and it forms a sheath around the visceral mass. And it also forms a mantle cavity, which in our mollusks is water-filled, at least for our aquatic mollusks. So not too terrible. Sometimes we have little structures that allow efficient circulation through the mantle cavity, right? So there's one tube, and then we might have a second tube. Oop, that's red. I wanted a different color. There we go. And one of these would be an in-current flow, and one of those would be an out-current flow. So X-current, in-current. So depending on what kind of mollusk you are, that's the mantle. All right, continuing on, the radula. I can't draw it much better than this very simplified image right here. It's just a muscular projection that functions as a tongue that has these mineralized rasping structures on them. So rasping, and I'll throw in quotes, teeth. And it's just for scraping. So scraping food. It's not present in all mollusks, right? The radula is missing in bivalves. So bivalves don't have it, clams and mussels. And also sea slugs. Sea slugs being a term that is sort of common language for nudibranch. So when you say sea slug, you're generally referring to a nudibranch, and a nudibranch is a mollusk. However, if you say marine sea slug or marine slug, you're referring to an annelid. So good times, good times, good times. Okay. Nervous system, right? Our nervous system is pretty simple in most mollusks. In some mollusks, it's very well developed. So essentially, you have the esophagus. And surrounding it is a little ring of nervous tissue. That's the brain. So all mollusks have a ring of nervous tissue surrounding the esophagus. The complexity of that ring varies based on the class. So, and then we are part of protostomes. So we have two ventral nerve cords. And I'll put brain in quotes. Nerve ring. Especially when you get into interesting organisms like octopus, the nerve ring around the esophagus is not the primary nervous structure. The arms also do significant amounts of nervous activity, including like interpreting sensory information. So, which is normally a brain thing. Like in humans, all interpretation of sensory information up here. In mollusks with more advanced nervous systems, that sort of information processing is distributed, which is pretty darn cool. So, two ventral nerve cords is just going to be present in all of protostomia that has nerve cords. So, it's a hallmark of protostomia. Okay, so reproduction in mollusks. Most mollusks have separate sexes, by the way. For you students in spring 2024, hour and 15 minutes. For the Monday, I will not be present. So reproduction. Most mollusks, let's go ahead and just give myself some space to write. Most mollusks have separate sexes. Whether that be male and female, or, hermaphrodite, so male versus female. A lot of snails and other marine mollusks are hermaphroditic, so hermaphrodites are also common. Kind of happens when you have a phylum that has over 93,000 described species. We could say most are separate sexes, but hermaphrodites are also very common. So fertilization. Generally external. This is because for mollusks, they are generally aquatic. So external fertilization is fine when you're just shooting your gametes out into the water around you. If the feller next to you shoots their eggs out and you shoot your sperm out, then things will work out in the water around you. It's going to be fine. All right. Some have internal fertilization. So a lot of terrestrial, mini terrestrial are internal. Snails have penises. And then cephalopods. Also internal, which is kind of fun. Cephalopods, in case you've forgotten, or in case I didn't mention it, octopus, squid, cuttlefish. So there we go. Now, in marine snails and bivalves, the eggs hatch and we get trochophore larvae. Trochophore larvae being this little basic larval structure here where we have kind of a band of cilia around the middle and then little tufts on either side and diamond shaped. If you have a question saying what kind of larva is this, you don't actually probably need to be able to recognize trochophore larvae on sight because it's like the only type of larvae where we specify what type of larvae is it. So, Anyway, in some, there's a secondary stage. So in some, the trochophore larvae is pretty much the free-swimming larvae. So trochophore in marine snails and bivalves. And then some have a secondary larval stage. The second larval stage, right? A trochophore is missing a number of the hallmarks of being a mollusk. So, what does the veliger stage have? Second larval stage is veliger or veliger. Who knows? I'm confidently stating veliger. And basically, what we have is the appearance of the foot, shell, and mantle. So, pretty simplified stage, but a bit more complex than your trochophore. So here is a veliger with a shell. And here's our foot. And then inside the shell, right, that's our mantle. Not too terrible. Okay. For mollusks that produce a shell, which is all of them, well, except I think the nudibranchs, but pretty much everything else, including cephalopods, produce a shell. It's the mantle that secretes the shell. So that's kind of neat. All right, let's get specific here. In Mollusca, we have chitons, right? We have gastropods. We have bivalves. And we have cephalopods. So for chitons, they're kind of pretty simple. So we'll just do a quick list of chitin features, right? They're all marine. So they are all aquatic, living in the oceans, saltwater. Let's see. This is going to get real difficult to see if I have to draw on drawings. So all marine chitons. They have 8 overlapping calcareous plates. Right? Their bodies appear segmented due to these overlapping plates. They give the appearance of segmentation. They are not segmented. Segmentation is specific to annelids, and also there's some segmentation present in our insects. So appearance of segmentation, but they are not segmented. Basically, their foot is for locomotion and suction. Right, so those 8 overlapping plates allow for, limited body movement. They provide the body some flexibility. And the other thing they allow them to do, if you look on the underside of a chitin, they're going to have sort of an indentation in the center of their foot. And so they can flatten out on a substrate and then they'll arch their plates ever so slightly, pulling in that indented area and creating suction. So, pretty cool. Creating that suction also creates a little reservoir of water for them. So they have a little bit of water, so they can survive being in intertidal regions and exposed to water when the tide is out. So that's kind of cool. Okay, gastropods. Gastropoda is our snails. Oh, shoot. Did I say slugs are annelids? I meant leeches are annelids. Leeches are the ones that actually say. Oh, jeez. Jeez. Well, I can't edit that out. Uh, I'll try and make a note of it in the actual thing. Point out that I'm a dum-dum. And I said slugs are. Anyway, okay, so gastropods. Snails. Oh, that was nice. Slugs, nudibranchs, and abalone. These are cool. They're the largest class. So Mollusca is the phylum. Gastropoda is the class. So class Gastropoda. They are the largest class. And you won't have to memorize it, but of the 93,000 species of mollusks described, roughly 40,000 are gastropods. So they're also the most spread out across multiple environments. So that's pretty cool. Very successful class of mollusks. All right, so let's talk about some basics. All right, in our generalized mollusk body plan, we have the mouth at one end, the anus over here in the mantle cavity on the opposite end, as you would kind of just expect were you to draw an animal like this. Gastropods are exemplified as having torsion. So, the mantle cavity and the anus have been moved anterior. Basically, one side of the mass grows faster than the other. So we have our visceral mass, right? And then our little critter is growing, but one side is growing faster than the other. And it's going to cause that side to twist over, right? And that's going to produce our little snail. And then that part of the mass will grow and grow and grow and grow, right? So this is the process of torsion. So if you're looking at a gastropod, the tip is the oldest section. They are not segmented, but the ones that secrete shells, they sure kind of look it because of the twisting shell. So anyway, that's kind of cool. So because of this, right, we have our mouth and we have our anus. Uh-oh, what's this? Mouth, anus. So essentially, beep, 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 beep. That drawing looks weird. There we go. Mouth. Anus. Torsion causes the mantle cavity to move anterior. Anterior, front, posterior, rear. So the mantle cavity gets essentially shifted anterior simply because it's part of the visceral mass that is growing faster, and because it's growing faster, it folds over, twists over into the front. So that's kind of cool. Anyway, so this is torsion, right? Torsion. To get all wordy about it, portion of visceral mass grows faster than the rest. Mantle cavity and anus, because the anus opens into the mantle cavity always, are twisted and moved anteriorly. They poop on their heads. All right, so that's torsion. Rotation up to 180 degrees. So what's kind of cool is in some of these, especially nudibranchs, they can feed on cnidarians and incorporate nematocysts into their body. So, no, they are not making their own nematocysts. They are generally munching away on sea anemones and then incorporating them into parts of their body as defensive structures. So here is the cnidosac at the tip of one of these little defensive tentacle-like structures. And that's pretty cool. Gives them a defense. This is a nudibranch right here. Also present in, so the marine slugs are nudibranchs. Anyway. Eyes are the other big thing. So we have eyes on a pair of tentacles. So they can retract the eyes or stick them out, give themselves a little bonus when it comes to being able to observe the environment around you. Helps you out if you do not have to raise your entire head up to look around. You can stay low and then just extend the eyeballs and look around. I wish I could do that. Okay, that's gastropoda. On to bivalves. Bivalvia. So, bivalves are clams' mussels. By the way, abalone I do not have a picture of them, but if you've ever had an abalone shell, you probably recognize it. It has sort of this cool look. It's got some holes around it, generally like that. And the inside of it is very shiny, like looks kind of like mother of pearl almost. It's got this metallic pearlescent sheen to it. Abalones are a type of gastropod. Most people would think they've got one half of the shell of a fancy clam. It is actually the whole shell of a gastropod, which is pretty cool. Less cool, abalone are threatened and endangered in their areas because, well, their shells are pretty. You know, how human of us. Well, that's pretty. Let's kill as many of them as we can. They'll become more valuable if there's fewer of them. Okay, clams and mussels. The bivalves. They often have eyeballs, which is pretty cool. Just getting ahead of myself. Some of these image-forming eyes can get really disturbingly like people-like, which is pretty hilarious. Okay, so my actual numbered features, right? So, Glochidia are a veliger life stage. So, if you'll recall, the veliger is the start of the shell, and the muscular foot and the mantle. That's the veliger. Why does this veliger get its own special term, the Glochidia? That would be because this is a parasite. So, the Glochidia is a parasitic veliger. Essentially, clams are one of those critters that have, I didn't draw it here, that have a siphon. So you have that mantle cavity, and then you have the siphons. One is incurrent. Oh, yellow's not going to work there. And one is excurrent. So what's going to happen is we're going to get a bunch of eggs fertilized inside the mantle cavity, and then they hatch into larvae, and then, you know, you got the little trochophore, sure, and then they become the veliger, and they get shot out by the hundreds and thousands of the excurrent siphon. So they get propelled away, allowing them to, you know, distribute downstream. Basically, they float through the water until they use these very nasty looking attachment structures. Whoop. Right, we've got hooks. We've got spikes. And they are a gill / blood parasite. So they attach to fish gills and they hitchhike for, 20 to 70 days, at which point they can disengage and hit ground. So not only does the excurrent siphon blow them out and distribute them into the water, and then the current carries them in the water, then they attach to fish, and the fish distribute them throughout the environment. And they are able to colonize multiple areas, and after about 20 days they disengage from the gills and they hit ground and they lose their parasitic attachment features and they just become a clam or mussel. So this is freshwater only, interestingly enough. Freshwater only. Pearls are formed in our mollusks, and they are only formed when some kind of foreign irritant gets between the mantle and the shell. So here in my veliger, we have the mantle here, and you would have some kind of irritant. Basically, the immune system secretes shell around it. And since it's not exposed to the outside environment, it's that innermost layer of shell, the mother of pearl, and it forms the pearl. So it is an immune reaction to an irritant. Pearl farms, they make a small non-lethal cut into mollusks and deliberately insert things like grains of sand in order to guarantee that there's an irritant in there. And then once they've had that irritant long enough that they know there's a pearl, they then fish them out and kill the clams and yank the pearls. So it kind of sucks for the clams. Pretty, but sucks to be a clam. Dams represent a significant negative effect on the environment. Dams are bad, right? Clams distribute larvae into the current, and dams stop the current. Another thing clams generally need is flowing water. So our freshwater clams need flowing water. So in these deep reservoirs where water flow is basically nil, they tend not to do well. Another problem with these deep reservoirs is that they lay down basically, upstream in the headwaters and all the way down, we're picking up sand, gravel, all, fine particles, silt, and it's all flowing downstream. And then it all hits the reservoir and it builds up sediment layers, right? Clams sit in a sediment layer and filter food. And if you've got sediment constantly being deposited on you, that's another negative effect on their environment. So dams prevent larval distribution. Reservoirs are generally crappy habitats for clams. And then they require flowing water so that they can have continuous nutrient flow, right? If you're in a river, nutrients flow from headwaters down and out, right? So nutrients collect and flow. Dams stop nutrient flow. And then the other big deal about dams is, here's your river and here's your ocean. And then you put a big dam up there. And then, somebody else wants a dam. And then somebody else wants a dam. And then suddenly, out by the ocean, the river runs dry. So you can, with enough dams and with enough aggressive damming, you can destroy these estuary regions out by the ocean. And the other thing is that if there's not continuous freshwater flow through here, then backflow from the ocean and the tides is going to hypersalinate that water. And if you're a freshwater clam and you get exposed to this hypersaline water, well, your cells shrivel up and you die. So, not a good time. All right. Here's my bivalve. There's my mantle epithelium as indicated. And then here's the beginning layers of the shell. Those initial shell layers secreted by the mantle. And then there's the mature shell. And basically, you get an irritant right here in that epithelial layer. And the irritant gets larger, and some of this stuff is immune system-wise secreted around it. And you can see as the shell grows, the tissue migrates and the pearl gets deep into the tissues, which is why collecting pearls is lethal. So, yeah. 10% of the 300 freshwater species of clams in the U.S. have gone extinct, in large part due to damming of river systems. A big one is the Colorado River. There was a Colorado River clam, the Colorado clam. And it is almost extinct. And the reason it's almost extinct is because it lived in the Colorado River Delta. Essentially, right here, where the Colorado River flows into that sea there. And nowadays, it's dry. And the water that does get in there is the backflow from the tides, and it's hypersalinated. And what you can find is massive dunes of dead clams dating back a century or more, right? So we can look at live clams and shells of dead clams from prior to the 1930s, prior to the Hoover Dam. And most of the rest of the dams along the Colorado River. And we can see that there's hallmarks of shells that indicate a lot more freshwater. Basically, they can look at the oxygen bound up in the shell, and they can see that it's associated with freshwater. And most of the Colorado River clams today have the oxygen in the shell that's a hallmark of saline, hypersalinated too. So they're not doing well. There are exercises in, so there are political movements trying to get regular opening of the dams to create regular freshwater flow through the delta, to also try and cycle a lot of the silt, sand, and gravel that builds up in the Colorado River that would normally be flushed out into the bay, or that bay there. Man, I am suddenly forgetting it. Anyway, yeah, bad time to be a Colorado River clam. The Colorado clam is not doing well, and that is due to damming. Okay, let's get into my upside favorite mollusks, the class Cephalopoda. The cephalopods. All right, so let's talk about that foot. That muscular foot has undergone heavy modification. What is the muscular foot? It is tentacles. The muscular foot has undergone heavy adaptation into tentacles. By the way, the tentacle itself is a very specific term. So what is a tentacle? It's not just that muscular structure, but a tentacle specifically has suction cups. And those suction cups, if you were to look at one in a little more detail, right? Let's see, try and get a little depression in there. Yeah, kind of working. They have a hook in them. So, suction cups are not only a little cup-like structure that allows for suction, they also have small hooks that allows for attachment. So, these little adhesive suctioning cups with hooks in them, and these are effective in prey capture and also fine manipulation. You know, the same stuff we'd use our fingers for. So they are capable of doing some very fine manipulation of their environment. They also have a lot of fine sensory structures on their tentacles. So they're very good at doing finger stuff. So what can octopuses do? Octopuses do a lot of finger stuff, which is pretty cool. So capturing prey, fine manipulation, that is what the foot has become. The in-current and ex-current siphons that you see in clams, there's something analogous here, except it's now a single structure. So if we're looking on the underside, of a squid. Here's my eyes. And here's a bunch of tentacles. This is my siphon. We no longer need, well, no longer. There are not two siphons here. And the reason there's not two siphons here is because we now have much finer muscular control. We can expand the siphon to suck water in. And then we can do a contraction of the siphon and the mantle cavity to push water out. So if you have your siphon, right, so here's my neutral siphon position. I can, expand it. And as I expand it, the volume in the siphon increases. And it basically, it creates a little water pressure thing. And that water pressure thing causes water to get sucked in. So, contract and expand the siphon. And then the other thing I can do, or not contract, relax. The other thing I can do with that siphon is I can contract it. And then I also, so here's my mantle cavity. So we got my mantle cavity. Mantle doesn't, this is conceptual, so I am exaggerating the kind of expansion you can get here. But then there we go. We contract and constrict the mantle cavity and the siphon. And when that happens, water shoots out. And you get a high velocity jet propulsion effect. So this is put to good use in a large number of cephalopods as an escape mechanism. So it's not necessarily just constant general locomotion. They have often adaptations for locomotion. In octopus, they do a lot of swimming with their arms. And then in squids and cuttlefish, they have various degrees of finnage along their mantle. And they can do basic swimming. Well, not basic, but they can do some, slower, let's say slower than jet propulsion swimming. Very delicate abilities to swim, especially when you're talking cuttlefish. They are highly maneuverable. A cuttlefish's finnage is almost along the entire mantle cavity, which is pretty cool. So they're capable of really fine degrees of movement. But then whether you're an octopus, a cuttlefish, a squid, or one of those little weird ones that doesn't quite fit anywhere, or the nautilus, which is super primitive and cool, whatever you are, you have a siphon, and you can fire water out of that and get some extremely fast movement, whether that's for escape or, just rapid locomotion, whatever. They have jet propulsion. Other cool features, chromatophores. Most have chromatophores. And basically, these are pigment cells. So here's a cell. And it has sacs of pigment. I don't want to get stuck here drawing forever. And those cells can expand and contract those pigment sacs. So here, we'll just expand one. So there's some blue that has expanded, and maybe also some green, right? Mix some colors in there. And we can also constrict. Right, so our chromatophore is a cell with pigment sacs that can expand and contract and that causes color changes. And these can be quite dramatic. These can play significant roles in not just camouflage, obviously camouflage. So cryptic coloration. They can assume a wide variety of colors and patterns to enhance their camouflage ability, but also one of the cooler features is communication. So these can be very complex signals, especially when you get into cuttlefish. These can also, there's some evidence that there's emotional expression, which is pretty cool. So, for instance, if you're a cuttlefish chilling out along here, and here is a cuttlefish. And let's say you are a male cuttlefish. This is a female cuttlefish. And then right over here is a competitor male cuttlefish. And what you can do is on this side, you'll display a mating display to attract a female. And on the other side, you will do female colorations. And you'll have female patterning on the side facing the male, the competitor male, and you'll have a mating display on the side facing the female. And this is another great example of April Fool's animal deception, which is pretty cool. So cuttlefish, a male cuttlefish will deliberately get its way between a competitor and a female in order to do this kind of thing, which is hilarious. And if you think about it, that kind of thing, it's hard to use the word intentionality when it comes to this sort of display, but it is certainly a higher order complexity behavior, which is very cool. And that brings me into the fact that we have chromatophores allowing rapid color shifts. Cuttlefish have these patterns aren't just static. They're often, they appear to be moving. Those stripes appear to be moving along the side of their body, shimmering, flashing, right? This is very complex, cool stuff, and it indicates a lot of higher order nervous capabilities, the emotional expression especially. This brings us to the fact that these guys have the most complex brain of all vertebrates. Most complex of all invertebrates. I think I just said vertebrates on accident. Of all invertebrates. That is pretty amazing. They are very high on the sentient spectrum, extremely high up on the sentient spectrum. Most people will often place them with birds like corvids. If you're not familiar, corvids are often placed up there with primates. So These are exceptionally intelligent organisms. So pretty darn cool. They also have an extremely advanced eye. They have the advantage. I said this 100 million years ago in lecture, but there's my pupil. There's my eye. There's my retina. And the light detecting parts of the eye are facing the light. And that means the nerves all connect back and go straight back to the brain. There's no blind spot. Vertebrate eyes, on the other hand, right? The light sensing part of the vertebrate eye is actually behind the back of the eye. And that means the nerves all come up into the eye. The nervous layer is kind of forming the back of the eye. And so in order to create a nerve to the brain, we create a blind spot. So arguably, cephalopods have a better eyeball than us. This right here is ye old nautilus. And when I say ye old nautilus, I should be pronouncing that the old nautilus, because ye is pronounced the. Look it up. Thorn. That is not thorn. Look up the letter thorn. Okay. I'm not going to spend too much time in a biology lecture on Middle English from the 1400s. But the nautilus is the most primitive of all cephalopods. For one, it has an external shell. Whereas the rest of cephalopoda has a shell that is under the mantle. It's also highly reduced. Like if I were drawing this cuttlefish, its shell would be under the mantle, and it would be about that big. If you've ever had a bird and you've gone and bought a cuttlebone, that's literally the shell of a cuttlefish. So, not a bone, a cuttlefish shell. So that's kind of cool. All right. Boom. Nautilus tend to be a lot more primitive in many ways. One, no suckers on their arms. So they have arms, not tentacles. No suckers. Now my pedantic brain is wanting me to separate the no a little bit better. No suckers. So these are arms, but not tentacles. Arms are not tentacles in the nautilus, which is kind of cool. They also have a much simpler eye. It is image forming. Basically, this is a pinhole pupil. But there is no lens. The lens of your eye is a focusing structure. And there's also not so much a cornea either. The cornea is the outer clear layer of your eye that is also a helpful focusing structure. This has the outer layer of the eye is not shaped to aid in focusing. We just have a pinhole. However, just having a pinhole eye still produces an image, right? Which is pretty cool. So they still have an image forming eye, even though it's very primitive. Basically, a very, very small pinhole still allows light to come through, and it still projects an image. This would be why, even though I have awful vision, if I do this, I literally cannot read a single thing on here. None of the words are visible. My vision is very bad. But if I form a very small pinhole with my hand, oh, look at that. Cephalopods, right? So I can read a lot more. So the very small pinhole structure of that eye still allows for limited focus, very small field of view, and yeah, still allows for an image. Nautilus tend to be deeper ocean organisms, very cool organisms. Okay, so now most can produce ink. Basically, they have a little ink sac that can secrete a very dark pigmented liquid into the mantle cavity, and then they can use their siphon, constrict their siphon, create that jet propulsion, and shoot out a cloud of ink to confuse predators. Not great if you try to keep something like an octopus, because their ink can make their water toxic. So it's built to, it's able to confuse and irritate predators and give them a chance to escape, because cephalopods tend to be very fleshy and not very resistant to teeth. So there you go. Pretty cool. Now, these cephalopods maintain a very high level of metabolic activity. They are extremely active. They can get large, very large. The colossal squid is a abyssal zone squid. Probably my favorite term for depths of the ocean, the abyssal region. Basically, that extremely dark water area just above the bottom of the ocean. They live in the abyssal zone, and they can get 20 meters plus, which is 66 plus feet in length. So that is a very large animal, right? They have extreme metabolic needs. Needs that can only be provided by a complete or closed circulatory system. They are the only mollusks with a closed or complete circulatory system. None of the other mollusks have a closed circulatory system. They have a heart, they have arteries, they have capillaries, they have veins that return the blood to the heart, right? And so there are no blood sinuses in these organisms. These have the advanced circulatory systems that you see in vertebrates. So that's pretty darn cool. Again, that would be convergent evolution, which is pretty wild, I think. So, lastly, reproduction in mollusks or in cephalopods. We are dioecious, 2 sexes, male, female, and and we do internal fertilization. Now, interesting, no penis, right? So, much like all other mollusks, the gonads are exposed to the mantle cavity, right? Which then has that siphon. And in males, what they do is they produce this sperm packet. It's a little packet packed with sperm, a little tissue enclosed sperm packet. And what they do is they have a tentacle that they reach in and they grab onto that. So here comes the tentacle, and it grabs its spermatophore, and then it's holding on to this spermatophore, and it basically just tries to put its spermatophore into the mantle cavity of a female. So I guess I drew the tentacle in green that time. So here's my mating tentacle. My mating tentacle. My mating tentacle. And it's just going to try and insert the spermatophore into the female's mantle cavity. Whoa. So the mating tentacle gets inserted into the female mantle cavity. And they deposit their spermatophore. And that is their big deal. That is the internal fertilization in cephalopods, which is pretty darn cool. So let's see, I talked about the internal shell, with the nautilus being the only ones with the external shell. Yeah, so that's this slide. Here we go. These are amazing pictures of what those chromatophores can do. So this one is a cuttlefish, and it is displaying some of that extreme communication ability. This is an octopus showing its ability to be cryptic. They also have muscles along their tentacles that can change the texture of their skin. So that's pretty cool. This video is being played in reverse, which is kind of fun. So this is in reverse just to show the dramatic difference between the camouflaged octopus and its environment. So It's a very quick response. Wild stuff that's not going to get tested on, but their eye is very well advanced, but it is black and white vision. They can't resolve color, which is deeply fascinating considering their bodies do so much with shifting color. And what they found is that basically the sensory structures on their body interact with the arms, and there's basically reflexes that cause the change in color and the change in texture. So they're changing color, but they don't see the color they're changing in order to be cryptic, which is kind of wild. So this right here is a mimic octopus. And it is kind of cool at creating silhouettes. So here is a silhouette that is similar to a flounder. Here's a silhouette similar to a lionfish. This is a silhouette similar to a krait or sea snake, Krait. And basically it can mimic other organisms in order to be able to serve different functions. Flounders tend not to be prey for a lot of the things that would hunt an octopus specifically. So they can do some predator avoidance that way. They can mimic a lionfish fin spread in order to take advantage of the fact that lionfish are venomous. Those fins are spiky and venomous. So it can do a little mimicry there. Same with the sea snake. Sea snakes are also highly venomous. And then this is a comic I really enjoy. Southeast Asian sea life. Mimic octopus, mimic octopus, mimic octopus, mimic octopus, mimic octopus, mimic, 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 mimic. This anchor, that's a mimic octopus. This submarine, definitely a mimic octopus. Here's a kraken, that would be 2 mimic octopus. Here's a school of fish, that's definitely a mimic octopus. And over here, that may look like a scuba diver, but that is certainly a mimic octopus. And then one of my favorite little videos.
And then there's this unsolved mystery.
Is a cuttlefish that is currently pretty well camouflaged in its environment. And here is a crab. So this is my camo. That's pretty good cryptic coloration. It is hard to see that cuttlefish. And there is a crab they've deposited in there, and it is going to hunt and eat that high-value food. Boop. Whoa.
And then there's this unsolved mystery.
Down. And
it changes color when it grabs a snake.
This is not camouflaged at all. That creates some very strong contrast with the environment it's on. So why, when grabbing the crab, did it suddenly lose all its camouflage? Well,
and then there's this unsolved mystery.
Glomp.
It changes color when it grabs a snack.
That doesn't make perfect sense because it seems to make it very conspicuous. So one theory is that, it's just a happy signal of how excited it is to have caught something.
An excitement signal, an emotional response. So that's pretty cool. All right, our cephalopod varieties. Nautilus. Mini arms. It does not have suction cups. So mini arms, a large external shell. Squid. Elongate, thin internal shell called a pin. They have smaller fins at the back of their body. They have arms, and then they have two long prey-catching appendages that have tentacles only at these front pads. So these are our prey grabbers. That's a thing of squids. Cuttlefish, they have W-shaped pupils. They have oblong internal shells. They tend to have better finnage. They have the little grabbers as well, but they tend to be smaller because cuttlefish tend to be smaller. And then octopuses. Octopi is not proper plural. They have 8 tentacles with suction cups all along them. They have little to no shell, and that means if it can fit basically their eyeball, they can fit through an opening. So the only thing that can't be completely squished through an opening is their eyeball. So pretty awesome. And that, my friends, is Mollusca. Mollusca. 4 minutes, 22 seconds left on our Monday equivalent for you spring 2024 folks. So take a second and chill, because next up is phylum Annelida. I'm going to pause this. I'm going to keep that 4 minutes because now I'm going to do an hour and 19 minutes to finish this lecture off. Well, fingers crossed we'll finish this lecture off. Okay. I'm going to chill here for a second so that if you're sliding that YouTube bar around, you can catch me doing this and know that this is the break point. That's probably long enough. Okay. For you spring 2024 folks, 1 hour, 19 minutes, and start. Phylum Annelida. Phylum Annelida can be broken up into 2 groups, the polychaetes and the oligochaetes. These are our 2 main groups of annelids. So let's start with Polychaeta. This is one that most people don't see a lot, even though it's the largest class with 10,000 plus described species. These are pretty much marine. That's what's going with the Polychaeta, and why a lot of people don't really see polychaetes. They don't think polychaetes. Polychaeta literally means many hairs. Poly, many, chaeta, hairs. The polychaetes have many hairs, especially visible in our Christmas tree worm. And our fun little critter here. Very cool. So, largest class of annelids. They have a very well-developed head. So, the prostomium, pro as in a head or in front, stomium is our well-developed head. right? We have eyes, we have antenna, we have sensory structures, often sort of meaty, flexible things. Sensory palps is an interchangeable term. Then we have parapodia. So phylum Annelida is segmented. Our body is broken up into segments. Boop, 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 boop. A feature of polychaetes is that they have appendages on most segments. So, parapodia, appendages on most segments. And those appendages tend to have those mini hairs for which the class is named, Archaeae. So, pretty cool. Our parapodia have a number of functions depending on what kind of polychaete we are. So they include a wide variety, including crawling for our polychaetes that chill on surfaces like the bottom of the part of the ocean they're in, right? Swimming for polychaetes capable of free swimming in the environment, and even attachment for polychaetes that need to anchor to structures such as this Christmas tree worm. It anchors to a home spot, secretes a tube, and then chills there. So it's parapodia. It will secrete itself a little tube. And then it will use its parapodia to hold itself in its tube. And then here's its prostomium. And here's some palps. These are modified for filter feeding. And so it can stick its palps out of the water to filter feed. And then if it sees something nasty, it will scoot itself back into its tube with its parapodia and hide. And it will just anchor itself in its tube. So that's pretty cool. But, nope. All right. That's pretty cool. The chaetae are bristle-like. So these are not hairs that are sort of thin and flexible, little flowing hairs. They're not human or mammal hairs. These are bristle-like structures. That's because they are made of chitin. Chitinous, which is the same stuff as the insect exoskeleton. That is a solid spelling of chitinous, chitinous. made of chitin, chitin. So for those that need to dig and anchor, those bristle-like chetae come in pretty clutch. All right, reproduction. Generally, they lack permanent gonads. When you see one of these chilling out, it probably doesn't have gonads. Fertilization tends to be external. So during breeding season, they grow a reproductive segment with their gonads. And when the time is right, they fire them gametes into the water around them. Sometimes they disconnect their reproductive segment with the gonads entirely, and that wiggles around in the water to help kind of distribute the larvae. So they'll like knock their their reproductive segment will disconnect and wiggle in the water column and secrete all its sperm or eggs as it wiggles around in the water column, which is pretty useful if your sperm and eggs are a highly nutritious food source, which they often are for a lot of fish. So if you want to prevent I just dropped my digitizer pen. But if you want to keep yourself from becoming a tasty treat because you waggled that sexy, delicious reproductive structure out in the water, why not fire your reproductive structure off like some kind of anime mecha shooting its fist out? And then hide. Hi, Oscar. There it is. Some polychaetes also reproduce asexually through budding, not nearly as dramatic, but pretty cool if you have a pretty good, stable environment and you don't need to shoot out all your gametes and distribute them in the ocean currents. Why not just bud off a new part of yourself and have your clone chilling with you and help reproduce your species that way. So not too bad. That's pretty cool. The Paula worm. This is not the Paula worm itself. This is once a year in November. A full moon hits and they fire chains of reproductive segments out into the water. So this is an epitoke. And that is the female reproductive segment. And it can get over 1 meter in length, as you can see in that thing. So that's the female epitoke. The male epitokes are much, much smaller. But basically, these epitokes wiggle and wriggle in the water, and they float towards the surface. And when there's enough light, basically they have a photosensitive reaction. When the moonlight gets bright enough, the epitokes explode and fire their gametes out. And now they're in the upper currents, able to be distributed in the higher currents of the ocean, which is pretty cool. Gotta love a reproductive strategy that involves explosions. literal gonads blowing apart in order to release gametes. That's pretty darn awesome. Okay, Oligoketa. Oligoketa. There are not nearly as many hairs on oligochaetes. Oligos literally means few. So chaete, again, cheta is hair, so few hairs. So this is earthworms and leeches. They all have chetae. What they don't have are parapodia. So parapodia, if you recall, are the little appendages on either side of a segment. So in a marine polychaete, as we illustrated, we have our little parapodia, and then the chetae are attached to the parapodia. Here, in my earthworm, our chetae are attached directly to the segment. No parapodia. No parapodia. Now, not having parapodia means you can't use your parapodia for locomotion. So, how do these organisms locomote? Well, the action is called peristalsis. Basically, they have along their body muscles that are perpendicular. So some muscles run vertically, and they can contract and make the animal constrict. So it gets thinner. That's the muscles that run vertically. Other muscles run longitudinally along the length, and they can relax and lengthen and they can constrict and shorten. So they can contract and shorten. And so basically, peristalsis is waves of this action, right? So you'll get a constriction and a relaxation that causes a small segment to push outward. And then these will dilate, the circular muscles will dilate and allow it to expand, and then it will constrict again. And if you want an analog to how peristalsis works, well, actually, if you want an identical type of muscular action, our esophagus, our entire gastrointestinal tract operates on peristalsis. We have concentric muscles, and we have longitudinal muscles. So when I swallow, right, above the food, it constricts and lengthens to push. Below the food, it relaxes and contracts. And then it moves a little bit, and then we constrict and lengthen, and it allows it to move. And that is going to be how food moves through the entire gastrointestinal tract. These are coelomates, so they peristalsis to move their body, and they peristalsis to move their guts, baby. So that's pretty cool. Oligochaetes are hermaphroditic So they're all hermaphroditic. The other thing is that earthworms have permanent gonads. So where are their permanent gonads? This sort of smoothed, larger segment called the clitellum. And basically, in order to mate, they get together and they secrete an extremely sticky glue. And they glue the clitella together. They glue those parts of their body together, and that actually facilitates gamete exchange. So rather than having to secrete their gametes just against each other and have the danger of them kind of being affected by gravity and losing a lot of them, they just glue themselves to each other so that they can mate very easily. So the clitellum is what's secreting that glue. So these are two earthworms glued together by their clitella, exchanging them gametes. The other neat thing is that they secrete a mucus cocoon, which acts as a protective covering for an egg. So if you were to look at that, if you're the type of person who buys earthworms as feeders, which I actually am, sometimes when you buy those feeders, you'll see these spherical yellow-orange, almost like little nodules in the dirt of your earthworm container. And that is the mucus coating that protects an embryo. Protective mucus coating. This means that you can see earthworm eggs and that they are surprisingly large, which is pretty darn cool. So this is a cocoon of sorts. So these eggs do not have a shell. They do not have a fibrous coating. It's not like the fibers that an insect weaves a cocoon out of, like a caterpillar. This is a mucus cocoon, but it's still pretty cool. It's protective in that it offers some protection from desiccation and predators. So that's pretty neat. Right. The chetae on earthworms, since they're no good for locomotion, because there's no parapodium in order to move those chetae around, basically they are anchors during peristalsis. Hey. Oh no. Well, this is about to crash. Pause. Oh, did we resume? We resume. Hooray, no crash. Resume. Anchors during peristalsis. So that's how annelids do. Now, leeches are a separate, have separate features. They actually used to put leeches in their own class before we had molecular biology to group leeches in with earthworms. So this is because leeches have some very distinct features to separate them. First of all, when talking about leeches, right? So here's my leech. There's my leech suction cup. There we go. While you'll often see little creases in their body, that is not separated segments. What I mean by that, if you look at an earthworm, right, very few things are continuous through their segments. The digestive system is continuous, along with its associated muscles, but other than that, these are kind of separated off. So, do. And these separations between segments, plural septum, or plural, singular septum, plural septa. And these create very distinct separated segments. And you'll see a lot of repeated organs in there. So, for instance, they tend to have these little structures that secrete to the outside that are basically kidney-like structures in that they serve a filtration function. Which is pretty cool because Annelida have a somewhat complex circulatory system. So one of my favorite facts about annelids is that if you're looking at their circulatory system, circulatory system is another one of those things that is continuous through the body along with the ventral nerve cord, because we are still protostomes. All invertebrates are protostomes. They have 5 dorsal hearts. If I were an annelid at Valentine's Day, I'd say I heart you. Earthworms just have 5 times the amount of love to give. So, back to leeches. So, they're not separated segments. They have no septa. Right? That's one of the features that made them separate leeches into a separate class from earthworms. Another one of the features, they have no chetae. None of these little hairs on their segments. So the segments are largely external in appearance, because without the septa, there's like it's truly a segmented organism. But how much can you say they're segmented if they're not like completely divided off? You know what I mean? It's the fuzziness of biology, right? Can you truly say they're segmented if we're not saying separated body segments? But then again, they have repeated structures along those undivided segments. So are they actually segmented, but just no septa? They used to throw these into their own class because of these differences. But then molecular biology showed genetics included them in with earthworms. So they're all part of class Annelida. All right. So there's roughly 500 species of leeches. And most are aquatic. So that's pretty cool. Most are freshwater. There are some aquatic leeches that are marine. There are some leeches that are terrestrial. Most aquatic freshwater leeches. So leeches, famous for parasitic feeding. They want to suck your blood. How they do this is that they secrete a natural anesthetic and a natural anticoagulant. So they secrete an anesthetic and an anticoagulant. An anesthetic reduces your ability to feel the wound that the leech makes and to feel the leech actually feeding on your blood. The anticoagulant, if you're not familiar with the term, coagulation is the term for the thickening and solidifying of blood to create a clot. So an anticoagulant prevents a clot from forming so that the blood can flow freely. Fascinatingly, they cannot directly digest blood. They have symbiotic bacteria. Actually break down the blood. So that the leeches can absorb the nutrients. So the bacteria break down the blood, and the leeches, they feed way more than the bacteria need. So the bacteria break down all the blood, take their fill, and the leech gets the rest. Leeches are hermaphroditic, and they all reproduce sexually. However, you might notice in the drawing of the leech, and also in this picture of a leech, there's no clitellum. Right? No clitellum visible. This is because the clitellum only forms during breeding season. At breeding time. So their clitellum is only temporally produced. So they produce their reproductive segment during breeding season in order to reproduce. And then once breeding season is over, the clitellum breaks down. So it's a real energy saver. Okay, continuing on into clade ecdysozoa. We are out of Lophotrochozoa. We have now completed Lophotrochozoa, the organisms with lophophores and trochozoan or trochophore larvae. So lophophore feeding appendages, like in the, what do you call them? Brachiopods. So they had the lophophores. And then also the trochophore, trochozoan larvae. So there you go. Now we are in clade ecdysozoa, our final clade before going on into deuterostomia. 46 minutes left in our Tuesday equivalent. Or not Tuesday, Wednesday equivalent for you spring 2024 folks. So what is the reason for separating these guys out into their own distinct clade? Why clade ecdysozoa? Well, that is because of the presence of ecdysis. What is ecdysis? Very specific. Ecdysis is the molting of an acellular, A Latin for without, so a non-cellular outer coating. That outer coating is secreted by the epithelium. It is honestly analogous to the shells in clams, right? Shells are not a cellular structure. They are a calcareous, hard protective structure. Here, shells, however, grow continuously and are never shed. Once you get that veliger that has its first shell, that veliger will be the original shell. If you see a clam, they have that hinge, and then the shell grows from there. That was the veliger shell, and then it just secretes as it grows, right? These are not shed. They are a permanent feature. Ecdysozoa, they molt their acellular outer coating in order to grow. Larger. Some molt an unlimited number of times. Some molt only a limited number of times. So, they molt in order to grow, and they molt an acellular layer. There are only two groups. Phylum Nematoda, the roundworms, and phylum Arthropoda. They have very distinct, different outer coatings that they molt. Hence the very general term outer coating. So let's dive into nematodes, baby. Roundworms are found in every conceivable ecological niche on the planet. Every niche, right? You have free-living roundworms. You have symbiotic roundworms. And those symbiotic roundworms, well, they can be found as parasites, as commensal, and as mutualists. In other words, you have the parasites that harm their host, as they benefit. You have the commensals, where the nematode benefits and the host is unaffected. And you have the mutualists, where the nematode and the host benefit from living together, right? So all niches. Soil can contain billions of nematodes per acre. So soil nematodes have billions per acre, right? There are 25,000 plus species of nematodes. Honestly, probably 100 times more than that, because again, when you're talking billions per acre in the soil alone, getting it, analyzing all these different nematodes to see if they're different species probably becomes an exercise in futility. That's way too many to spend all your time. You could spend a lifetime just looking at different nematodes in one field, right? And identifying new species. So, all right. Many nematodes are indeed parasites. There are 50 plus human parasites. So they have medical and economic significance, especially in the parasitic ones. So what is their acellular outer coating that they molt in order to grow? What is the acellular coating that sits them square in ecdysozoa? It is a cuticle. Uh-oh. Oh no, Panic at the Disco. There we go. Got it going again. The acellular layer is a cuticle, right? So this over here is a nematode, and we've cut away its cuticle. So this is the cuticle. And then it's impossible to tell what the digestive structure in there is, because it's completely obscured by reproductive organs. Basically, on either side of the digestive system, they'll have coiled, packed in there reproductive structures, especially the ones with ovaries. Tons packed in there. So there we go. Ascaris lumbricoides is an intestinal roundworm. So Ascaris lumbricoides is one of the most common worm parasites in humans. They have a life cycle where they mature in the intestines. So they achieve sexual maturity in the intestines. And so they lay their eggs into poop. The embryo matures in the soil. And then due to poor sanitation, like say, animals that we, slaughter for food, are cruising around, kicking up dirt around their own poop and getting those inactive eggs containing an embryo, right? They get them all over their body, and then during slaughtering, some of those eggs remain, right? And cooking sterilizes the eggs, right? But if it's undercooked, you swallow the eggs. The larvae will then The eggs will then go vegetative. The larvae are released into the intestine. They penetrate into the intestinal wall and into your vessels. And then they move through the vessels to the lungs. They break into the alveoli of the lungs. And they grow during their embryonic stages. They can only molt a limited number of times. So limited molts. Adults do not molt. So adults do not molt, so they have a limited number of molts during larval development, which is occurring in the lungs. And then they migrate through the alveoli, up over the trachea, and back into the epiglottis, into the stomach. They can do that thick outer cuticle. They can survive the stomach acid and get into the intestines, and they complete maturation in the intestines and sexually or asexually reproduce there. So an infected human can crank out 200,000 eggs per day. So that is a high degree of fecundity, which I realize is a word you haven't heard before. So fecundity. It's just slang for basically the amount of eggs and offspring you crank out. A highly fecund species cranks out a crap load of eggs and larvae. So high degree of fecundity in these guys. So Ascaris lumbricoides, big deal. What's the rate of infection? Up to 64% of people in the southeast U.S. end up with an Ascaris parasite at some point in their lives. And as much as 20% of the entire world population has the Ascaris infection. In addition, we have hookworms, pinworms, dog heartworms. Elephantiasis is a condition you can get from a nematode infection. So here we have a mosquito. And basically, the mosquito, there's a nematode parasite, a little filarial nematode. And the larvae migrate to the capillaries of the outer layers of the skin. So we have little capillaries. And larvae go into the mosquito's gut, and they mature in the gut. And then when the mosquito feeds on another person, whoop, nematodes. This is elephantiasis. And this is terrifying because this is tissue filled to bursting. Like, it's not actually bursting, but it looks like a water balloon that's about to pop. So it's not like your skin actually explodes, but it's filled with nematodes, right? There's inflammation and then just billions upon billions upon billions of nematodes. If you cut into the skin, you would see so many nematodes. Oh, so many nematodes. It has got to be insanely gross. Okay. Now, sucks to be a, human if you get a nematode parasite. However, there are nematode parasites of pest insects. So there are parasites of crop pests. And that means what you can do is take nematode eggs and larval nematodes and basically just aerially spray crops. So crop dust with nematodes. And that can help you control crop pests. So that's pretty darn cool. Basically, what's happening is that they have bacteria that are GI tract, so intestinal symbiotes of our nematode parasites. And essentially, the nematode penetrates the exoskeleton it migrates into the insect, the body cavity, the coelom, if you will. And then it secretes a toxin that inhibits the immune system. So it secretes an immune system, immune system inhibitor. And then that also releases the bacteria into our insect. And then the bacteria secrete digestive enzymes and toxins that are what actually kill our pest and allow digestion. So this is a crop pest, a beetle larva that has been killed with nematodes. So that is the roundworms. And now let's rock 30 minutes into Arthropoda. So Arthropoda is the most successful in terms of number of raw number of species, right? There are more than 1 million species. 2/3 of all animal species are arthropods. That's a lot. So they are of extreme economic importance. Again, positive and negative, right? So, pollinators. Arthropoda, a lot of Arthropoda co-evolved with our angiosperms in order to pollinate them, right? That's amazing. So many crops are pollinated by arthropods. So many crops, right? So in addition to those really nice positive qualities, there's more than 1 million species, right? So we're also going to have agricultural pests. And we're also going to have a lot of parasites. And a lot of those parasites and various others can spread disease. So while it looks like I have more negative economic factors than positive ones, honestly, pollinator is Such a huge positive factor that it outweighs everything else. So this is our other segmented invertebrate. However, it does not have repeating segments. So its segmentation is different than the segmentation in annelids. Annelids have repeated segments. Arthropods have distinct, different segments with a distinct head. So we have a head. We have a thorax, and we have an abdomen. So in general, your head is where we're going to have various sensory structures and complicated mouth parts. The thorax is where we're going to have our appendages. Legs, wings, etc. The abdomen right here is where we're going to have part of the GI. So not the entire gastrointestinal system that runs through it, but a lot of what's analogous to the intestines is in here. So intestines, rectum, damn near killed them, and the anus. We're also going to have the gonads. In some organisms, sometimes respiratory structures. So a lot of aquatic organisms have respiratory structures, whether they're tubes or something like that, sticking off of their abdomen, and they can breathe through their butt segment. So that's kind of cool. In fact, the rectum has a ton of capillaries, so sometimes their rectum is modified that they can extend a portion of their butt out of their butthole and just breathe through there. And they can just inhale into their rectum. So, that's one way to do it. Okay, so this is their segmentation. Speaking of appendages, they have jointed appendages, and this is one of the keys to their success. So, their appendages have multiple joints and modifications. So, jointed, modified, specialized structures. So, what counts as jointed appendages? A lot. Okay, so complex structures, generally very flexible because of those joints. They allow a high degree of movement, locomotion. You'll have adaptations for, movement and attachment. Right? If you see little beetle feet, beetles have little, almost fuzzy, jointed structures at the end. So there's a little beetle foot, and that helps them move. And these little microscopic filaments down there help them, well, not microscopic, but the little pointy bits help them cling to stuff and climb on stuff. So that's pretty cool. A lot of spiders, instead of having multiple pointy bits, will have an end segment that is like a single very pointy bit. And then at the microscopic level, it may have a bunch of little hair-like structures coming off of it, not hairs, and those hair-like structures create a massive amount of surface area for clinging to stuff. So adaptations for movement and attachment, but that's not all, folks. Locomotion is but one. So locomotion, legs, wings, mouth parts. They often have complicated mouth parts. This allows for generalized feeding. They can be generalists. So if you look at a praying mantis, it's got a mouth that's very roach-like. So it has bits that can open up to allow stuff in. So this is lateral movement. And then in addition to those lateral parts that can open up, it also has these vertical parts that can open and close. So it can chew from all directions. And then even then, it has multiple little pointy bits inside there. So it can chew laterally, vertically, and then it has little pointy bits that can grab and pull. And so when a praying mantis grabs prey, the prey will be alive and it will chew its legs off. And then it will chew on its head to a certain degree. Sometimes they don't eat its entire head, and then they just chew it open and eat whatever they want. And they are very good at eating a wide variety of insect prey. And then you also have a lot of specialized mouth parts. Think mosquitoes that have hypodermic needle mouth parts. There are assassin bugs that have a thick, hardened mouth part. Well, a mosquito mouth part is very flexible. An assassin bug has a mouth part that is very long and pointy, and they keep it folded against the ventral part of their body. And then when they come up on prey, they whip that out and just stab it through the exoskeleton. So they can prey on armored arthropods. So that's pretty cool. A lot of them have claws, grippy stuff, something you might not realize are jointed appendages. Antennae. So they have a wide variety of sensory appendages. So that's pretty cool. Locomotory appendages, complicated mouth parts, sensory structures, right? Bam, very cool. These jointed appendages are a huge part of their success. So, boop, 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 boop. They have, in the realm of ecdysis, their exoskeleton. So, their exoskeleton is basically our acellular outer layer and it's made of proteins and chitin. Chitin being very similar to the cell walls of plants in that it's made of beta glucose. It's just a lot more branched, which is pretty cool. So lots of proteins. This makes it a very rigid structure, but also it can have flexible joints. So can be flexible. Think about at the joints and anywhere else where it's thin. So thinner layers of the exoskeleton allow for a lot of flexibility, which is pretty cool. Thicker layers provide rigid armor. So that, again, very cool. All of their appendages are exoskeletal, right? There's muscle associated with it that's attached to the inside. But yeah, this is the exoskeleton, our hard outer layer that is molted. So this is what gets molted, which again is why it's in ecdysozoa. Right? Pretty cool. So if you have your jointed appendage on the inside, you'll have your muscles attached to the jointed appendage to allow for various complicated movements, right? So they can contract their muscles. So muscles attached to interior surface is muscle attachment. To the inside, of course, nothing has muscle on the outside. Big part of this, protection. Obviously, protection from predation. Also, protection from desiccation, drying out and dying. A thick cuticle provides a pretty effective surface to prevent water loss. So, exoskeletons put a limit on body size. They are comparatively heavy. So, gravity affects maximum body size. In addition, they have limited respiratory structures. So, limited respiration. And so diffusion of O2 through the exoskeleton. Diffusion from essentially holes in the exoskeleton, right? That also limits size. You have oxygen limitation. we can breathe quite a bit in, and then our hearts pump it very efficiently. Speaking of, arthropods have an open or incomplete circulatory system. So, again, another limiter on oxygen distribution. So, between the heavy exoskeleton, the fact that we have limited respiratory structures, that the respiration has to occur at essentially holes in the exoskeleton that limit its protective abilities and limit its protection from desiccation, means that oxygen largely, there's very limited ability to pump oxygen into a body of an arthropod, and so that limits size. So because of diffusion and limitations on the amount of holes they can truly have, and then they have that open circulatory system. So not too bad. Exoskeleton, hard muscle attachment protects, and the disadvantage, as we said. 10 minutes to go. I think we can keep going through this slide. Open circulatory system. We already hit that one. Ecdysis, right? They're shedding their hard exoskeleton. Now here's the thing. New exoskeleton is soft. In other words, before their exoskeleton hardens, man, they are extremely vulnerable to predation. Extremely vulnerable to predation. Again, ecdysis and exoskeleton production has a high metabolic cost. Energetically expensive to keep this exoskeleton going. So these are limitations to these guys, to this strategy. All right, let's talk about respiration. So, as I said, they have to respire through holes in the exoskeleton. There have to be pores in the exoskeleton. Those pores are called spiracles. So they're the holes or pores in the exoskeleton that allow gas exchange. Remember how there's limited pumping and oxygen distribution? Well, they have tracheal tubes. So these have muscles. So there's muscles associated with the tracheal tubes that allow them to relax and widen, and then contract and constrict and do limited O2 pumping. So they can do some gas pumping. But again, it's kind of low efficiency. So not super efficient unless you're particularly small. Smaller, more efficient. Also, this is a big thing for our terrestrial arthropods. Now, some have primitive lungs. So arachnids have book lungs. So a little specialized structure for respiration. It allows arachnids a greater ability to both distribute oxygen and resist desiccation or drowning. Right. So because they have most of their respiration concentrated into these book lungs, as long as the book lungs have access to O2, they're going to be fine. A cricket, where most of its spiracles are on the ventral part of the abdomen and the side of the abdomen, if a cricket hits water, it drowns very quickly. Even if it's half submerged, it's going to drown. So, yeah, not great. So, and then gills tend to be pretty effective. So they are more efficient. So if you have book lungs or you have gills, you're going to have more efficient respiration. And if you're a smaller arthropod, your spiracles and tracheal tubes are, as is the evolutionary goal, good enough. Okay, so the nervous system. There is cephalization. After all, they have a head. So there are cerebral ganglia. I'll put brain in quotes. The reason I'm putting it in quotes is because having a concentration, a small concentration of nerves in the head is certainly making us think of a brain. It's analogous to a brain by location. Now, again, these are invertebrates. They are part of Protostomia. So we're going to have a little nervous ring around the esophagus. Hey, look at that. Analogous to the cephalopods or in the mollusks. So that's kind of cool. And then we have our ventral nerve cords. Now, we're going to have accessory ganglia, right? There's going to be more ganglia. So there's ventral. So the anterior ganglia are our cephalized ganglia, but there's ventral ganglia. And oftentimes, those ventral ganglia are predominant processing structures. Essentially, the cerebral ganglia, which we would think of as a brain, are mostly controlling the head attachments. They control head appendages. But not so much behavior. They don't tend to control the behavior of the organism. That's ventral ganglia stuff, which is pretty darn cool. Okay, so as far as the excretory system, excretory system, right? This is in the form. So first of all, excretory system. Think filtration, urination. like in humans, the analog would be our kidneys and our, ureters to the bladder, and then your urethra to your external urinary meatus, the opening that your urine comes out of, right? Okay, so in arthropods, most have Malpighian tubules. And these are our excretory systems. These are projections from the digestive tract. And essentially, they filter out nitrogenous wastes. Examples being uric acid and guanine. Now, big deal of having an excretory system is that you can concentrate waste and conserve water and salt. That is a big deal. And so this is a key adaptation for getting onto land, for terrestrial life, right? Because desiccation is a big concern. And if you have to ditch a lot of water in order to get rid of your nitrogenous wastes, you're not going to do very well on land. And in a lot of organisms, in order to get rid of your wastes, you have to excrete salt into that waste. And the Malpighian tubules help conserve salt. So, pretty cool. And guess what? For you spring 2024 folks, welcome to the end of Invertebrates Part 2. That is 2.5 hours. So, all right, 10 slides left. So, spring 2024 people, you're going to get these slides in class. But I have no problem with making this lecture one of the ones I have online that I can offload students onto if I am running way behind. Again, online lectures are why I don't have real homework. There's no mastering biology portion of my Bio 2 section, and that's because occasionally we have to do the online lectures. So instead of making you do like 30 minutes of homework twice a week, you get to occasionally take notes from an online lecture. But hey, I'm an entertaining guy. So once again, spring 2024, bail unless you want to have some reinforcement of what I'm going to go over in class. So we are on slide 22, and let's continue with key adaptations in Arthropoda. Okay, so first of all, some arthropods have ocelli. This is what we refer to as a simple eye. We can see them on this praying mantis. And basically, they detect light versus no light. You'll often see ocelli on the top of the head. That would be because a lot of predators are the type that hunt from above, birds especially. So if a predator is going to do death from above, that also means that they're going to have a shadow. So ocelli can detect a shadow, help you escape. Also, ocelli are useful in horizontal stabilization, especially during flight. So now let's talk about the super fancy insect eye, the compound eye. So what is a compound eye? So this is present in many crustaceans, and of course the class derived from crustaceans, insects, and also in centipedes, and in the extinct trilobites. And basically, it is an eye made of visual units. So here is essentially a lens that focuses light. And that is a visual unit. And then you just interlock hundreds to thousands of visual units. And then in order to take advantage of these visual units, see this praying mantis? It kind of looks like it has pupils. But what it actually has is a jointed retina. And so inside this eye, this little jointed retina is going to be going, what's that? What's that? What's that? What's that? And it can move its retina around in there. So it almost has a little eye stalk, not truly an eye stalk, but it has a little movable receptor thing. And so it can change what it is focused on simply by moving the retinal appendage within the compound eye. So that's actually super cool. So a big deal in crustacea, which is where we find insects. And then also in certain centipedes. And we can see their evolutionary origin in trilobites. So there we go. That's our other big non-universal key adaptation. You would be, if you were just going by compound eye, you'd end up with paraphyletic groupings, which is kind of cool, depending on what your level of focus is. So that's kind of cool. All right. So let's go on into our breakdown of Arthropoda. Right? Whoa, hey, that is not what I wanted. Boop. First stop, Chelicerata. What is a Chelicerata? This includes spiders. So This is what you think of if someone says arachnids. So arachnids is kind of an older term. I use it because, well, most everybody knows what an arachnid is. However, in phylogenetics, you would not use arachnid. It's kind of like pachyderm. If you were young enough, and went to the zoo, there was the pachyderm house where they had the rhinos and the elephants and the tapirs and the hippos, and it was a catch-all term for extremely large herbivorous animals, mostly from Africa. And as it turns out, as far as a classification term, it is a garbage term. It does not include related organisms, right? Rhinos, elephants, hippos, extremely diverse in terms of evolutionary relationships. So arachnids kind of gone by the wayside as far as full-on pedantic phylogeny goes. Now we have Chelicera, Chelicerata. Arachnid. Oh, jeez, I keep using it. So, spiders, scorpions, ticks, mites, the daddy longlegs, which are not spiders. So, roughly 57,000 species. Most are terrestrial, some aquatic, but the grand majority terrestrial. Okay, so what is Chelicerata named for? First off, when someone says arachnid, you think 8 legs. 12. Because we don't really use the term legs, we use appendages. 6 paired appendages. Come on. 6 paired appendages. Ooh, things are getting crowded. Eight walking legs. 4 pairs of walking legs. 1 pair of pedipalps. What are pedipalps? If you've ever looked at a spider, especially a jumping spider, right up by their so-called fangs, they have these little, what's that? What's over here? I'm looking around. And a lot of jumping spiders communicate by drumming their pedipalps against surfaces. Boop, 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 beep, 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 beep. Anyway, Chelicerate Morse code. So that's pretty cool. They can be for manipulation. But they can also be for reproduction. So, for instance, in spiders, especially jumping spiders, but I think all spiders, they will collect their sperm in a packet with one of their legs and then pass it over to their pedipalps. And then the pedipalps are for insertion, so into the gonadopore. So there you go. So multifunction. cephalized. So appendage, anterior appendage. How's that? We'll get as general as possible. And then their namesake, Chelicera. So this is a pair of fangs for feeding, or pincers. 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 Okay, so what are other features? The thorax and head are fused. What do we call it? The cephalothorax. What else? What else? So they have a fused thorax and head, then they have an abdomen as normal. So, unlike the basic arthropod body plan with a head, thorax, and abdomen, they have a cephalothorax and abdomen. So, these two body segments are referred to as the tagmata. So, they're two body segments. What else? They lack antennae. And they lack mandibles. What are mandibles? Remember my whole drawing of praying mantids? They have rah, 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 and meow, meow, meow. Those are mandibles. So, specialized feeding, crushing mouth parts. Those are all mandibles. Chelicerates do not have mandibles. So, if you see chelicerate, just a little bit, that is a member of Chelicerata. So, spiders are venomous. And their fangs inject venom. But that's not only what they inject. They also inject digestive enzymes. So the venom is incapacitating their prey, possibly even killing it. And as the prey is, incapacitated, sometimes not enough to kill it, but just enough to prevent it from being able to move, their insides are then turned to liquid slurry. And the chelicera of your spiders, being modified into hypodermic fangs, can only take liquid into them. So they suck their liquefied gunk in there. So that's pretty cool. Fun fact: there's no such thing as a native brown recluse in Colorado. Indeed, every brown recluse in Colorado has either been found to not be a brown recluse. The absolute overwhelming majority of brown recluses are not brown recluses. House spiders, wolf spiders, all sorts of different spiders are just called brown recluses. You could take a picture of a small dog in a costume and someone would identify it as a brown recluse. The few brown recluses that have been identified in Colorado are literally brown recluses that moved with someone from an area where brown recluses are native. So if you moved from the southeast U.S., and there was some brown recluses amongst your boxes, that's how you get a brown recluse in Colorado. But there's no established wild breeding populations of brown recluses. So that's pretty cool. Spiders have ocelli. Now, some of them are actually beyond light versus dark. You can have multiple eyes. Some are like they have image forming capabilities. Jumping spiders are famous for being able to analyze topography. Fun experiment with jumping spiders. They had a little spider in a little tower where it had a little closed door, and then they had a little block environment with little bridges and stuff, and then a delicious, delicious bug at one end. And they would open the door, and the spider would see the whole thing and analyze it, but it wouldn't be able to cross it like a little window. And then they'd close the door, and then they'd mess with it. And then they'd open the door, and the spider would crawl out and move through it. And where things got changed or messed with, the spider would stop and sort of react, not sort of like with a little bit of confusion. It would stop. It might crawl up something to observe things. And what this all boils down to is that spider would not be confused if it didn't have a memory or mental image of the environment it observed. So that's pretty complicated right there. Jumping spiders, man, adorable. Okay, so there is such a thing as a poisonous spider. Poisonous is you eat it and die. Venomous is it bites you and you die. So there are poisonous spiders in like temperate and subtropical regions. They're going to be like super brightly colored to advertise that. But most spiders venomous. Well, not most. All spiders are venomous. Not too many of them actually have medically significant venom. Yeah. Done with Chelicerata. Scorpions, by the way, very cool. Their chelicera are their claws. Pinchy, 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 pinchy. So that's pretty neat. In order to eat, they rip their prey apart in order to have very simple mouth parts able to take the liquefied prey, their soft insides. Whee! Time for Myriapoda. So, subphylum Myriapoda, or Myriapoda. That sounds cooler and smarter. Let's say Myriapoda. Ooh. Myriapoda, including classes Chilopoda and Diplopoda. Centipedes and millipedes, respectively. Okay, general characteristics of myriapods. They have a cephalized head with sensory appendages, including antenna. So, eyes, Sensory appendages. Centipedes generally all have antennae. Millipedes often do not. But when present, sensory appendages up there at the head. Then they have separate sexes. Oh, sorry. Their bodies are repeated segments. However, we do not have like septa or anything. These are not annelids. They have all them jointed appendages and stuff. So repeated segments. This is often where we'll find our locomotory appendages. Right, we have separate sexes, separate male and female. That is a very excellent spelling of female right there. They have internal fertilization. They are egg layers. Ooh. And that is all of them. No budding and no hermaphroditism here. So, centipedes, Chilopoda. All right. The chilopods have one pair of legs per segment. One pair of legs per segment. Oh, you'll never guess what Diplopoda has. All right. The other thing is that they are all carnivorous. Man, I have poison all through here. They are venomous. So, these are their venomous legs. So they're venomous claws, which are modified legs. These are not chelicera, because chelicerates have chelicera, even though these are very similar to chelicera. But they are venomous fangs, and they can be medically significant. However, not your general house and garden centipedes. So, if you see a centipede in your house or out in your garden, don't freak out. Don't kill it. That is just there to be your friend. They hunt soil pests. They are voracious roach eaters, too. So, house centipedes, if you have a centipede in your house, mwah, your house is being kept clean by that little friend. So, nice. Don't get rid of them. Acknowledge your roommate. Give them a thumbs up. Some centipedes get very big. This is a cave centipede gripping on to a cave ceiling with its back legs, and then they hang in order to hunt. And every once in a while, an unlucky bat gets close enough that they snag it with their fangs, their claws, and inject venom, and then they eat the bat alive. So, your standard terrifying stuff. Okay, so let's look at this centipede right here. Here's two separate segments, each with one leg on either side, one pair of walking legs. Oh, but what would happen if these were to, oh, I don't know, fuse into one segment? You'd have two pairs of walking legs? What? Each body segment is a fusion of two segments. The tagmata. So they have two pairs of walking legs per segment. They are obligate herbivores. Well, most are herbivores. The grand majority are herbivorous. You have some detritivores. You rarely have insect eaters. Would you call that carnivorous? Fuzzy. Insectivorous, that's extremely rare. You're talking like teeny tiny, not quite so microscopic, but darn near insects. They tend to have pores between in their segments. And these pores are for defensive capabilities. They can emit defensive chemicals. A variety of chemicals. So many of them are stinky. Bad, no, stinky, stinky, stinky. Some of them are a little more choose violence. So some centipedes can emit irritants, and some have been known to emit cyanide gas. This is a defense against like arthropod predators. If you're much larger than an arthropod, you do not have to worry about the amount of cyanide gas they're capable of emitting. So, Diplopoda, two pairs of walking legs per segment. Lemurs in Madagascar have been seen picking up centipedes and not killing them, but knocking them around to piss them off, and then rubbing the centipedes into their fur. And basically, it's a decent insect repellent. And in some millipedes, they choose them. Not so, like in some millipedes, the chemicals they rub into their fur are not insect repellents, but they are psychoactive. So it could be lemurs use it as an insect repellent and as a way to get high. So that's pretty hilarious. Okay, on to everybody's favorite crustacean, insects. Okay, so we have Pancrustacea. And here are the crustaceans. And we have all our variety of modern crustaceans. And also coming off of crustacea is our hexapods, insects. Insects are crustaceans. Hard pill to swallow, probably why they created clade pancrustacea, so they could say, the insects and crustaceans. But probably within a few years to a decade, we might not have pancrustacea anymore, and we'll just have crustacea, and nested in crustacea, the insects, hexapoda. So this is the largest class of animals on Earth. So pretty darn cool. If you counted up the number of insects alive, 10 to the 18th. So that is a ton of insects. That is a number that is mind-bogglingly difficult to visualize, to internalize. If it was 10 to the 18th meters, it would be 100 light years. So if the number of individual insects alive at any given time were actually like meters, like if my grandma was a bicycle, but anyway, it would take light 100 years to go that many meters. So lots of them. Okay, let's talk about our insects. 3 distinct body segments. The head, the thorax, the abdomen. We talked about these basic body segments. They have one pair of antennae, and it is cephalized. It's on the head. They have 3 pairs of walking legs. And then they have 2 pairs of wings. These are not homologous to walking legs. In other words, these are not modified legs. So the wings did not evolve as a pair of legs that were modified into wings. This is not a branches becoming leaves situation. And of course, they are analogous to vertebrate wings. Just to remind you of our terminology, they are not a homology in which they share a common evolutionary origin. They are analogous, in which they serve a similar evolutionary function, flight, but they are not at all derived from the same evolutionary history. So not too bad. So, some we have a secondary loss. In other words, all insects had wings, right? Wings were the dawn of our hexapods, but some no longer have wings. If wings are part of the OG evolution and then a species of that lineage does not have wings, we consider that a secondary loss of a feature. So, fleas and lice are insects that do not have wings. Now, weirdly, in some, they have an extremely basal lineage. Little outgroup. And you have like the silverfish. And silverfish straight never evolved wings. So while we consider wings a key adaptation to insects, silverfish are a very weird exception, and probably a few other exceptions out there where their lineage just never developed wings. One of those fuzzy biology things I love so much. Wings are a key adaptation of insects, except when they're not. Okay, so two pairs of wings, you may ask. What about all the insects I see that don't have wings? Well, let's say you have a beetle. And that beetle, right? Here's my beetle. Boop, boop, boop. His abdomen is covered up. But when he flies, he only has one pair of wings. I should probably give him some antenna. Now we can see the thorax and abdomen. Anyway, beetle only has one pair of wings. What's up? I thought you said two pairs of wings. Well, what's this? Modified wings that are protective. So in beetles, the forewings are protective covers. So when we have two wings, we have wings that are closer to the head. There's my head. These are the forewings. And then we have the hind wings. So, in beetles, the forewings have become hardened protective coverings. In a lot of flies, they only have one pair of wings. What gives there? Well, these are halteres, and they serve a balancing function, and they are modified hind wings. I believe this is a crane fly. So, its halteres just happen to be very large, which is helpful. So, there you go. They all have two pairs of wings, even when in beetles they're modified into a protective coating elytra, which is just a cool term. You know, if you're writing a fantasy novel, pretty solid name for a character. Elytra! Use your healing magic! And then in flies, we have them halt ears. Okay, so insects have a type of cell called a fat body cell. You are not insulting an insect when you refer to the fat body cell. The fat body cell is much like fat in humans. It's nutrient storage. Fats can be metabolized to produce ATP. But interestingly enough, the fat body cells also perform detoxification functions. And the detox part especially means we have something analogous to liver functions. So fat body cells are not only for getting us fat bottom beetles, but also giving us liver functions. So that is a key adaptation of insects as well, unique to hexapods. Woo! All right, 30 of 32. Another feature of Arthropoda. Most undergo a process called metamorphosis, in which you have a distinct and morphologically different larval stage. And metamorphosis is the transition from larvae to adult. So the transition from larval form to the adult form. So, not all of them undergo complete metamorphosis. Complete metamorphosis, the caterpillar becomes the butterfly, right? In which case we have a larvae. And so here, complete metamorphosis. The egg is laid, you have a very distinct morphological larva. That larva feeds until it has enough nutrients in storage and enough growth to undergo complete metamorphosis. What is complete metamorphosis? It forms a pupa, which is a term distinct to complete metamorphosis, right? They pupate. In the pupa, they liquefy. They become a soup. And then during pupation, they reform into adult. Weird, wacky things about this. You can do animal learning tests with caterpillars. You can indeed train a caterpillar in small ways. And when you train a caterpillar, and then it undergoes pupation, and literally all its body parts liquefy into soup and then completely reform into a very different adult stage, uh, it retains those training memories. So that's kind of wild. All right. Now let's talk about incomplete metamorphosis. Okay, so first of all, complete metamorphosis also has our super fancy Latin term, holometabolous. Holometabolous metamorphosis. Holo literally means complete, and metabol means change. 88% of insects are holometabolous. That's a lot. Now, we can contrast this with incomplete insects. Now, you'll never guess, but roughly 12% undergo incomplete metamorphosis. What is incomplete metamorphosis? The larval stage is distinctly morphologically different from the adult, but not as dramatic as you'd think. The larval stage is mostly distinct in like coloration and size. It's a miniature adult and it has no wings. So, all right. It basically just goes from miniature to adult, and its stages in which it molts are called nymphs. Nymphs molt, and they molt a limited number of times. Before adulthood. And the number of different molt times, depending on which molt they're at, they call these instars. So first instar is out of the egg. Second instar, third instar, fourth instar, adult. They don't all have the same number of instars. Okay, so hemimetabolous. I'm gonna die. Let's get some water. Examples of hemimetabolous insects. Grasshoppers. Grasshoppers are hemimetabolous. Cicadas. Hemimetabolous. Mantids. Praying mantises. Hemimetabolous. Baby mantids are little tiny praying mantises. They're very cute. So when first out of the egg, they don't get wings, but they actually do get wings a little early. I don't know. Anyway, they're pretty cute. Mayflies, stoneflies. Dragonflies are hemimetabolists, although their larval stage is aquatic, so it tends to look very distinct. It's a lot more morphologically distinct than miniature adult with no wings, so that's kind of cool. Complete complete metabolism, like butterflies and moths, right? So instars is stages. You can also use instars for complete metabolisms if there are a limited number of larval moltings. So basically, instar is a term for a limited number of molts. So if the larvae or nymph has a limited number of molts, it has instar stages. That's just literally it. So, larval stage for complete, pupa, adult. That's holometabolous. The pupa is sometimes called a pupa, sometimes called a chrysalis. Chrysalis is for butterflies. So the chrysalis is this hardened pupate stage. So for instance, beetles, when they pupate, they do not form a chrysalis. They form just this hardened little weird kind of crappy looking pupa. Like it's vaguely pupa shaped, but it's like hardened into a protective sealed structure. So it is not a chrysalis. The chrysalis is where we truly have liquefaction and reformation. A pupa does not necessarily mean that we're liquefying in there. There's just large changes. Okay, 31 of 32 slides. On to Crustacea, the class, the clade that insects are nested in. So, Crustacea. Examples: lobsters, crayfish, shrimp, crabs, water fleas, copepods, barnacles, an awful lot. Almost all are aquatic. What are the constraints? Why are almost all of them aquatic? They have no waxy cuticle. No waxy cuticle on their exoskeleton. So if you look at an insect, especially an insect, because they tend to have a softer abdomen, their ventral abdomen, where it's softest, will have a waxy coating, a waxy cuticle. Other problems: crustaceans have gills. And then most have aquatic larvae. So these are our constraints that restrict most crustaceans to being aquatic. What is a key adaptation of crustaceans? One, they are the only arthropods with two pairs of antennae. All right. Tagmata. Like the arachnids, the head and thorax are fused into a cephalothorax with a distinct abdomen. Unlike the other arthropods that have the 3 segments. There are appendages on the abdomen. This is rather distinct. This is unique to the crustaceans. Appendages on their abdomens, and appendages, obviously the thorax and cephala. So the cephalothorax obviously has appendages. The portion of fused cephalothorax, that's the head, has, antenna and mouth parts. Those are a bunch of jointed appendages. The thorax has the walking legs, and for those of them that have claws, claws. And then the abdominal appendages are generally for swimming. So, swimming appendages. Now, centipedes and millipedes being segmented and having a large number of repeating segments, one could make the arguments that centipedes and millipedes have abdominal appendages, but where does the abdomen begin? And a little fuzzy on those guys since they have repeated segments. Okay, so Tagmata, fused head and thorax into cephalothorax, and then a distinct abdomen. Appendages, paired antenna, 2 pairs of antenna on the head. We have compound eyes in many. Right? We tend to have our walking legs, and then in some they have pinchers, and those pinchers are not chelicera. Because chelicera are in chelicerates. And then the mandibles for chewing are actually very complicated, fully jointed appendages. So they have much more complicated mouth parts, and they are fully jointed rather than just being, like in my praying mantis, we had the mandibles that, are lateral, and they basically have a little muscle attachment here that allows them to move back and forth, and then they have the mandibles that are vertical. And while that may seem complicated, they are not segmented. So this mantis it has non-segmented mandibles. These are much simpler mouth parts. Crustaceans, their mouth parts are jointed appendages. And even when the joints are highly reduced, they're still jointed appendages. So, yeah. Looky there. Looky there. We're done. Crustaceans do not have any kind of fangs or anything. That's why they're not chelicera, even though pinchers in crustaceans and pinchers in scorpions are analogous. Scorpion pinchers are modified chelicera, and uh, crustacean pinchers or modified legs. So now we're done. Now we're done. Congratulations to anyone who was not spring 2024 and sat through this whole sucker.